Hello everyone, this lesson is going to be on protein synthesis, and I'm actually going to split this up into two different lessons. It's kind of long if I do it all in one lesson. The two different stages or phases of protein synthesis, as you can see here, they are called transcription and translation. So although in this first lesson I will touch on both of them, it's primarily going to focus on transcription, and then we'll leave translation for the next part, the second part of protein synthesis. What you've already learned at this point is about the DNA, the structure of DNA. You've learned about the replication of DNA prior to cell division, whether it is mitosis or meiosis. But we need to keep in mind that what DNA is really just a set of instructions or a recipe in terms of how to make different things. And usually what we're talking about in terms of making, well, is proteins and protein synthesis. So when we think about a number of different activities that are carried out by cells, that are carried out by your body, just to give you a few examples, your red blood cells that transport oxygen. It's not the DNA that's transporting the oxygen, it's the protein hemoglobin that's transporting the oxygen. Your muscles, when they contract, it involves many, many different proteins. It's not the DNA that's responsible for the contraction, it's proteins like actin and myosin. The digestive system, all of the different digestive enzymes, they are proteins. Of course, we've learned about the endocrine system. We know that most of the hormones, they are protein hormones. And with the nervous system, we talked about various different membrane proteins. So the channels, the sodium channels, the calcium channels, the chloride channels, so those ones, and along with the sodium potassium pump, those are all these proteins that are involved in the generation and the transmission of an action potential. So my point is that it's proteins that are carrying out all of these different functions inside of your cells. It's not the actual DNA. Just to make that a little bit more visual, nice picture here where we see three different deer, and clearly one of them looks a little bit different. And this is the result of, yes, ultimately it is the DNA. The instructions in the DNA are a little bit different in the white colored deer compared to the two, what we'll call normal colored brown deer. And as we'll see, this is a result of a mutation to the DNA. And as a result of this mutation, there is a different protein or no protein that is produced in terms of coloration in the white deer compared to the darker pigmented deer. So yes, it is the DNA that provides the instructions, but what we actually see, so whether it's visual here or whether it's biochemical, in other words, the phenotype of that organism really does come down to the protein and not the DNA, at least not directly. So we talk about DNA as being the workhorses of the cell. They are responsible for all of those different activities that I mentioned, along with, of course, many, many other ones. Pretty much everything associated with your cell and the functioning of your cell in your body that you can think of will be associated with proteins. So I did say that I'm going to chat a little bit about both of these processes of transcription and translation. So we know about DNA, but what we haven't really said too much about is this one here, RNA. So we'll see that RNA is a little bit different, whereas DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So in order to make a protein, yes, we do need to use the DNA, but the DNA is found inside of the nucleus, and proteins are made. Protein synthesis occurs not in the nucleus of a cell, but in the cytoplasm. Unfortunately, DNA is big and bulky and it cannot leave the nucleus and go into the cytoplasm. So how can we make the proteins in the cytoplasm when the actual code, the genetic code, is inside of the DNA? So somehow we need to get a message from the nucleus into the cytoplasm, and that is the role of RNA, or at least that one that I'm going to primarily talk about here, which halfway down the page, it is called the messenger RNA, or simply abbreviated with a lowercase m in front, mRNA. So it is mRNA that is going to take a set of instructions, not all of the instructions in the DNA of the nucleus, but only the instructions that are necessary for the production of a particular protein. So when we use this word here, gene, gene is a set of instructions on a discrete location within one particular chromosome. And those instructions are made up of the A's, the T's, the G's, and the C's. And those are the instructions that provide the, well, instructions to make a protein. But in order to use this code, we need to get that information into the cytoplasm, and that is really the role of the messenger RNA. Take only the information for that particular protein that needs to be used or made, and move that into the cytoplasm. So transcription, this word here, transcription, refers specifically to that process of using the DNA, reading the instructions in the DNA, and making, as we will see, a complementary messenger RNA. Although this will be for another lesson, again, I'm going to have to touch on this, translation is then when the messenger RNA that is now inside of the cytoplasm, it can be used in order to convert it into a protein. Here it has it as a polypeptide. Polypeptide is just a whole bunch of amino acids all linked together. So again, you have a polymer, in this case, a polymer of amino acids, and that is what we call a polypeptide, and that is really what a protein is. And as you probably already know, the protein factory within the cytoplasm, they are the ribosomes. So when we do get on to protein synthesis, we'll need the messenger RNA, we'll need the ribosomes, and there will be some other things as well eventually that we will also have to talk about for that process. So in molecular biology, they do refer to what is called the central dogma of molecular biology. And it goes like this. We take the instructions in DNA, we read those instructions to make messenger RNA, and then the messenger RNA is used to make a protein, and the proteins are the workhorses again of the cell. Just to keep in mind where these different processes are taking place, and we're going to focus on the eukaryotic cells, so they're cells that do have a nucleus. We already know that DNA replication takes place within the nucleus, and what it shows very clearly here is the first part of protein synthesis, transcription, that takes place inside of the nucleus. So here we have our double-stranded DNA. From that double-stranded DNA, a portion of it is going to be read, and here it has it as pre-mRNA, and we'll talk a little bit about the distinction between pre-mRNA and this one here, which is the mature RNA. So a little bit of RNA processing that takes place. And then the RNA, it is smaller than the DNA. It's single-stranded, as we'll chat a little bit more about, compared to the DNA that is double-stranded. And it is small enough that it can go through these little holes in the nucleus, and they're called the nuclear pores. DNA cannot lead through the nuclear pores, but messenger RNA can. And then it's inside of the cytoplasm, where now we would have the second part of protein synthesis, translation, that's taking place. And again, we can see here that in addition to the messenger RNA, we would also need a ribosome and again, a few other things for that process. So we also talked a little bit about the history of DNA, the structure of DNA, going back to James Watson and Francis Crick in 1953, coming up with that double helical anti-parallel structure of the DNA, made up of the 4 different nitrogenous bases that we abbreviate with the A, the T, the G, and the C. Proteins, however, are not made up of nucleic acids. They are made up of something else, and that, of course, is amino acids. So somehow we need to take one kind of code, the code of DNA, and convert that into a different kind of code, the code of proteins, which is amino acids. So how does this actually happen, that the code in the DNA provides the instructions for making proteins? So whereas there are 3 nucleotides in the DNA, there are 20 different amino acids. In fact, there are many, many more than 20 different naturally occurring amino acids. But kind of interesting that biological organisms, and it's not just us, but all biological organisms, they only use 20 and exactly the same 20 amino acids for all of the proteins that are eventually made in all living organisms. But again, to make 20 different, or to make proteins from 20 different amino acids, we only have this 4-letter code in the DNA, the 4 different nucleotides. So how is that possible? How does this actually result from one kind of language, the language of nucleic acids, being converted into the language of amino acids, And this question here, which was a big question in the 1950s, is how many bases correspond to an amino acid? So is it one single base codes for one amino acid? Is it a 2 to 1, a 3 to 1, a 4 to 1 ratio? No one really knew, and there was a great deal of speculation for a number of years, and it did take a number of years before they actually pieced all of this together to figure out that, well, it is a 3 to 1 ratio that we're talking about. So this is a really nice picture here, and again, for this one, I am going to have to go into the part 2 of protein synthesis, which is translation a little bit as well, without getting into too many details. But starting at the top, what we do have is our double helical DNA molecule. Now keep in mind that DNA is really, really, really long. So this particular DNA, it's going to be going on for millions of bases in this direction and also in this direction here. So this is just focusing on a very small portion of this chromosome, of this DNA. So on any given chromosome, there might be hundreds or thousands of genes. And this one, we can see it's only identifying 3 different genes. And if we take a look at the second picture, it's saying, okay, let's just kind of focus on this, what they're calling gene number 1, without telling us what it really is. And let's just take a look at an even smaller portion, not even the entire gene, the set of instructions for making the protein. Let's just take a look at a section of that gene, a few of the bases that we do have here. So in the next picture, we do have our DNA template. And this is really important. The DNA, of course, is double-stranded. So we do write this, in fact, if I can try and write this using my mouse pad here. So DS does stand for double-stranded. So our DNA is double-stranded, as we already know, complementary double-stranded DNA. But out of those 2 different strands, in order to make a protein, it's only 1/2 of it that's actually being read. So what this picture is showing us is that it is the top strand here. So if I read it across, the ACC, AA, ACC, and so on, that is the coding strand. The bottom strand, which of course runs in the opposite direction, it is not coding for a protein, at least not reading in that same direction as the one at the top. So we're going to use that information. Remember that this information is restricted to the nucleus, but the protein is going to be made in the cytoplasm. So what has to happen through the process of transcription is we need to take that coding template strand of DNA and we need to make a complementary copy. So where we have the ACC, AA, and so on, we need to insert into our new messenger RNA that's being made the complementary bases. But if we take a look at our messenger RNA that is achieved through the transcription process, you'll notice that, well, there are 4 different bases, but there's no T. In DNA, we have the thymine. In RNA, there is no T, there is no thymine. So there are still 4 bases. What we have instead in the RNA is a U, which stands for uracil. So now if we don't have a T, what is complementary to the A is going to be the U. So starting with our template strand, we have an A in the DNA, so the complementary base would be a U. We have a C, the complementary base would be a G, and then we just continue on to the right with all of the other complementary bases. So if you see a T, you should realize it cannot be RNA. If you see a U, it cannot be DNA. So that's one of the differences between the DNA and the RNA. The other one, of course, is that DNA is double-stranded, but the RNA is going to be SS, which stands for single-stranded. Now, that is not always the case. Not all RNA has to be single-stranded, but this kind of RNA, we're going to talk about a couple of other kinds of RNA as well. This kind of RNA, though, the one that provides the instructions to make the proteins, the messenger RNA, it is single-stranded. So now let's go back to that ratio that I was talking about. And what we now know is that the ratio is a 3-to-1 ratio. So for every 3 bases that we do have in the RNA, that's going to result in 1 amino acid being inserted. So a really important term that it has down below our messenger RNA here, and it is a codon. When you see the word codon, it is only talking about one thing and one thing only, and that is 3 consecutive bases in RNA. It is not 3 consecutive bases in DNA. So if I take a look at, for example, these 3 bases here in the DNA, TGG, I can call that a triplet. Tri meaning 3, there are 3 of them together. I can call it a triplet, but I cannot call it a codon. Whenever you see the word codon, it is referring specifically to the messenger RNA. And again, as we'll see, that is actually kind of important. So if we take a look at these 3 bases here, the UGG, it just so happens that that turns out to be the code for this amino acid here. This is the 3-letter code, TRP, for the amino acid tryptophan. This one here, UUU, is the 3-letter code for the amino acid phenylalanine. GGC is the 3-letter code for the amino acid glycine. So how do we actually know what 3-letter code is for which amino acid? And again, this is the sort of thing that it actually took them quite a few years, like in excess of a decade, to piece all of this together. And now what we have is this wonderful, which is called the messenger RNA codon table. Messenger RNA, it can only be used for messenger RNA. Again, this is kind of jumping ahead to the next part of protein synthesis on translation, but just to kind of give you a heads up, this is how it works. What we do have is 64 possible 3-base codons, but only 20 different amino acids. So some codons, here we have 2 different codons that actually code for the same amino acid. Here we have 6 different codons here. Oops, all of these ones, they all code for the amino acid leucine. Without getting into too many of the details right now, if we just take a look at this one, UGG, quite simply, this is how you read the code. You find UGG right here in this table, and it tells you, well, yes, that is the amino acid tryptophan that's going to be inserted. One more time, I cannot stress enough, this table is only for messenger RNA. You would not use this for DNA, and you would not use this for what we're going to talk about in the next lesson, which is transfer RNA. This is only for messenger RNA. So through this process, again, this 3-to-1 ratio, we're taking the messenger RNA and we're making a protein, and that is the process, again, of translation that we're talking about for that. Okay, so enough with the translation for now. Let's go back and really kind of focus on transcription for the rest of this lesson. So the genetic code, of course, we're kind of focusing on humans and what's going on in humans, but it is universal. So, you know, it doesn't really matter if you're some other animal, if you're a plant, if you're a bacteria, an archaea, a protist, a fungi, they all use exactly the same genetic code, DNA, the A's, the T's, the G's, and the C's. So what that also means is it's, well, the same process, essentially, whereby this information in the DNA is transcribed and translated into a protein. And what that also means is if we're all using the same code, it's, well, interchangeable, essentially. So we can take a gene for a particular protein, a particular trait. We can take it from one species and we can transplant it into another. And we can also cross the species barrier and even the kingdom barrier. So what it's showing in this picture here, some kind of strange pictures, I suppose, but the one on the left-hand side, what we do have is a gene from a plant, a firefly, or sorry, from an animal, a firefly gene. And this is the gene that allows this firefly to glow in the dark. And what they did is they inserted into a plant. So not only are they crossing the species barrier, they're crossing the kingdom barrier. They're going from instructions that you would find in an animal and putting that into a plant. But you know what? Well, it works. So now we can see that the plant is actually glowing just like a firefly would. And the same thing on the right-hand side, only in this case it's between 2 animals, taking a jellyfish gene and transplanting it into the genome of a pig. And now we can see that, well, sort of like a jellyfish, we have the snout and the hooves that are glowing in the case of the pig. So really no practical application, of course, to this sort of thing. All it really does is show that this sort of thing is possible. But in the future, we are going to see that there most definitely are some real practical applications to transplanting genetic information from one species and into another. So what exactly is going on with transcription? What are the details behind how this messenger RNA is going to be made? Well, in some ways, if you think back to replication, it's kind of like replication. We need to open up the DNA, we need to read the bases, we need to bring in the complementary bases. And we saw that with DNA replication, it's a fairly elaborate process that is involved. Many different enzymes, 4 of which you guys needed to remember. So those enzymes were the helicase enzyme and the primase, the DNA polymerase, and the ligase enzymes. For transcription, fortunately, in some ways it's a little bit easier. It's easier in the sense that there's only one enzyme now that you need to know, and it is called RNA polymerase. So whereas DNA polymerase brought in complementary DNA nucleotides and created a polymer by linking them all together, RNA polymerase is going to create another polymer, only now they're just different nucleotides. They are RNA instead of DNA nucleotides. So the RNA that is made, again, there are some similarities and there are some differences, some of which we've already seen. So yeah, single-stranded in the case of RNA. And here I do have usually, so again, messenger RNA is always going to be single-stranded, but we're going to come across some other forms of RNA that are double-stranded. And RNA, yes, it has a different base. It has the U instead of the T, and it is the U that now would be complementary to the A. So I'm going to play for you a little bit of an animation here. So this animation is going to compare the DNA to the RNA. So again, we've already talked about the DNA. We already know the basic structure of the DNA, and this is going to review some of that, but also kind of focus on now the RNA and the differences between the DNA and the RNA.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, so that's a nice review of DNA and a nice comparison in terms of similarities and differences between the DNA and the RNA that we're kind of focusing on now. So the next couple slides, we're going to get into the process that is involved with transcription specifically. So the picture that we do have at the top here, what it is showing is, well, it goes by a couple different names. So the traditional name that we use for the coding section of the DNA that's responsible for making a protein is a gene, but that's starting to change a little bit simply due to the fact that a gene does have a specific definition, and that definition is that for each gene there is one single protein that is made. And what they're now finding is that's not always the case. And again, we'll chat a little bit more about that. So here they're using the term transcription unit. So I'll use the terms interchangeably, transcription unit and gene, but just think of it as a section of DNA along the length of the chromosome in a very, very specific location on that chromosome that provides the instructions for the making of one, or now we'll have to say one or more than one protein. So the darkly shaded DNA, that is the coding region of the DNA that's ultimately going to be transcribed to make the messenger RNA, and in turn that information used to make a protein. In front, so at the left-hand side here, this is in front of the transcription unit or the gene. It is read in this direction here, going toward the right. So in front is what we call the upstream region, and the upstream region before the actual gene or transcription unit is a section of DNA that is called the promoter. So it's the promoter where the one enzyme that you need to know about, the RNA polymerase, is going to recognize, and the promoter just kind of gives the RNA polymerase a heads up and kind of says, this is it. This is where you need to start prying apart the DNA, reading that DNA template strand, and making a complementary messenger RNA strand. So we can see in the next picture here, that's exactly what our RNA polymerase is doing. We can see now that the DNA, it has been separated, and that exposes the template strand. In this case, we can see that the template strand is the one that's at the bottom, and that is the one that's being read to begin the making of our messenger RNA. So this RNA polymerase is now going to move along the length of the DNA. As it moves along, notice that in behind, that DNA just snaps back together again. Again, those two strands, they are complementary to each other, so they do want to come back together. And what's being released in behind is our single-stranded messenger RNA transcript. So over at the right-hand side, they're just showing it in a little bit more detail what is taking place underneath the RNA polymerase, the enzyme responsible for this. Again, unlike replication where we needed these different enzymes involved, RNA polymerase is kind of doing all of the work on its own. So it's untwisting the DNA, it's prying apart the complementary bases, it's exposing the template on one of the strands, it's bringing in the complementary RNA nucleotides, and in behind, it's going to be releasing what we can see at the end here, which is our entire messenger RNA transcript. So now it's released. Not only is it released, as the DNA also just looks exactly the same as what it did in the first place. So this can then be used over and over and over again whenever more messenger RNA needs to be made, whenever more protein needs to be made. This is showing the same thing, just a larger view of what's actually taking place underneath that RNA polymerase, bringing in of the complementary bases. I'll show you 3 different animations here that are just going to walk us through the different stages, and these animations have broken it up into these 3 different stages that they're calling the initiation, the elongation, and the termination. So this first one here with our initiation, of course, they're going to talk about the RNA polymerase. They're also going to talk about something called transcription factors. So the DNA is a little bit more than just passive information that's sitting there waiting to be read. It does actually respond to cues within the cell and from outside of the cell as well. So how does the RNA polymerase really know this is the time to read the specific instructions, because this is the time to make more of a particular protein? Well, that information kind of directing the DNA that it's time for that information to be read, that comes from transcription factors. And we'll hear a little bit about those in this first animation here.
Part 1, Initiation. The promoter signals where transcription begins. RNA polymerase recognizes and binds to this site. The promoter is a sequence of DNA nucleotides, 15 to 300 base pairs long, that serve as the site where an RNA polymerase binds. In eukaryotes, a TATA box is typically present in the promoter, consisting of a nucleotide sequence like TA, TA, A. The TATA box aids in the recognition of the promoter. First, a group of proteins called transcription factors locate and bind to the TATA box. The transcription factors are a collection of proteins that help RNA polymerase recognize and bind to the promoter. Now, RNA polymerase II is able to bind to the promoter. RNA polymerase II is the enzyme responsible for transcribing the pre-mRNA strand. Aided by transcription factors, RNA polymerase II locates the start point and begins to unwind the DNA helix. Now, RNA polymerase II can begin transcribing the DNA sequence.
Okay, so now that we've pried open the DNA, we're just going to have that RNA polymerase moving into the actual coding section where we do find the transcription unit or the gene, and bringing in the complementary messenger RNA nucleotides. So let's take a look at the next one here, showing what they call in this animation elongation.
Part 2, Elongation. RNA polymerase II assembles the RNA nucleotides that complement the template strand. Just as in DNA replication, the enzyme reads the template strand in a 3' to 5' direction, while building the new RNA strand in a 5' to 3' direction. RNA contains the nitrogenous base uracil instead of thymine. Like thymine, it is complementary to adenine. RNA polymerase continues its path along the template as the DNA strands join back together in its wake. Let's zoom in and see this process in more detail.
So finally, what we need to have is the messenger RNA that's actually being released from the DNA after the template is being read, and that'll be the last one here on termination.
As RNA polymerase continues along the transcription unit, the newly synthesized RNA strand separates from the template.
Okay, so we didn't completely see it separated there, so let's just take a look at one other animation. This one doesn't have any narration that goes along with it, so I'll kind of walk you through with this one. So we're starting with our DNA. It's not twisted in the form of the double helix. We're just taking a look at the unwound DNA. So somewhere within here, we're going to have our promoter, and if we take a look here, this would be the TATA box that we have. So promoter in this region here, information is going to be read going to the right, and all of this is going to be the coding section of the DNA that we need to read in order to make the messenger RNA. So here's our RNA polymerase that's coming in. It's going to take a look at, it's going to recognize that promoter. Here they're not showing any of those transcription factors, but there would be transcription factors involved as well. There we go with separating the DNA, bringing in the complementary RNA nucleotides, messenger RNA being released in behind, DNA snapping back together simply because those are complementary bases, and that is what they want to do. And eventually we reach the end of the coding region, which is the terminator region. And now that messenger RNA is going to be completely released, so is the RNA polymerase. DNA looks exactly the same as when we started in the first place, and now we have our, again, shorter than the DNA, single-stranded as opposed to the double-stranded RNA, and it's this RNA that can now leave the nucleus through the nuclear pores and make its way into the cytoplasm, where the rest of protein synthesis is going to take place. However, there are a couple of modifications that are made before that actually occurs. So this picture here shows what is identified as pre-mRNA at the top, and then what we would call the mature messenger RNA at the bottom. So in the case of our pre-mRNA, in fact, what we do have is a number of sections within the DNA that are not going to be necessary for the coding of the protein. Well, let me just get my pointer here, my pen instead of my highlighter. So sections that do code for the protein, they're what are referred to, they're referred to as the exons. Ones that don't code for the protein that eventually need to cut out, they are called introns, which stands for intravenine sequences. So within our pre-mRNA, we have all of these RNA nucleotides. We have a polynucleotide, but what this is saying is that, well, we need to cut some sections out and we need to join the remaining sections together. So those sections that are joined together, we talk about splicing, and they are spliced or joined together. So that then creates our mature messenger RNA. What we can also see is these yellow protective regions at either end of the messenger RNA. The ends need to be protected because when the messenger RNA goes into the cytoplasm, there are in fact enzymes, they're called exonucleases, and they tend to chew away at the ends. So essentially, to buy a little bit of time and read as much of this coding information and make as many proteins as possible, you need to protect the ends. So before the messenger RNA goes into the cytoplasm, what happens? Cut some sections out, join the remaining sections together, protect the ends, and now it can go out through the nuclear pores. So this is really kind of significant as well. So I mentioned that we're kind of getting away from using this term gene because the definition of a gene is a section of DNA which is responsible for the production of one single protein. But what we now know is that that is not always the case. So from this, and instead of gene, I'm going to call it transcription unit now. What we have is this long strand here, which is made up of green sections, which are the introns, and the reddish sections, which are the exons. So this is what we now know can happen from this one single section of coding DNA, this transcription unit. We can take different exons and splice them together. So because we can take different exons and splice them together in a process which is called alternative splicing, what we can get is, well, not just one. Here it shows two, but it can even be more than two proteins that are produced from one single section of DNA. And this is the result of this process here, which is the alternative splicing. So that's it for, well, the first half of protein synthesis, which is transcription. So the rest of this here we'll leave for the second half, which would be our translation, and that'll be for another lesson.